Thanks for joining us in another fascinating journey through the world of science here on the Science Channel podcast.
Great to be diving into another topic.
Absolutely. And hey, if you're enjoying these explorations into, well, the nuts and bolts of life, please do hit that like button and subscribe. It really helps support the channel.
Definitely. Every bit helps.
So today we're plunging into a really intricate world inside our cells. We're talking about mitochondria, the powerhouses, right?
That's them. Essential energy factories.
But specifically, we're looking at how they make their own proteins. It's this whole separate system kind of tucked away.
It really is quite remarkable. They've got their own DNA, their own ribosomes. It's a whole different setup compared to the rest of the cell.
Yeah, and it's not just some minor side project for the cell. The system makes these 13 super important proteins, very hydrophobic ones, apparently.
Core components for making energy through oxidative phosphorylation. Without them, the cell's energy supply pretty much collapses.
And we're finding out more and more that glitches in this mitochondrial protein production, they're linked to all sorts of things, right? Diseases, aging.
Absolutely. From specific genetic disorders right through to potentially contributing to the aging process itself. It's a critical system.
So today's deep dive, we're using a comprehensive scientific review article to really unpack the control mechanisms. How do mitochondria regulate this vital protein synthesis?
Exactly. We're going to look at how it starts, how it keeps going, how it stops, and some of the really clever regulatory tricks mitochondria use.
Okay, let's start with the basics then. Mitochondrial translation, what's the big picture overview?
Right. So at its heart, it's the process of taking the genetic info stored in that mitochondrial DNA, the mtDNA, and turning it into functional proteins. It's the final step of mitochondrial gene expression.
Okay.
And while, mitochondria evolves from bacteria and shares some history, their translation system has really adapted over time. It's specialized for making those 13 specific, mostly hydrophobic proteins needed for the OXPHOS system.
So it's tailored for the job. What are the key components inside the mitochondria that make this happen?
Well, you've got the mtDNA itself. Small, circular, double-stranded DNA. It codes for those 13 proteins, but also the ribosomal RNAs, the rRNAs, and the transfer RNAs, the tRNAs, which are essential building blocks for the translation machinery itself.
So it provides the instructions and some of the tools.
Pretty much.
Yeah.
And interestingly, transcription often produces these long RNA strands, polypeptide transcripts, that contain multiple genes. These then get processed into the mature messenger RNAs, the mRNAs.
And some mRNAs carry instructions for more than one protein.
Yes. 2 of them are bicistronic, meaning they code for 2 proteins. And sometimes the coding sequences, the open reading frames, or ORFs, even overlap. It's incredibly efficient, packing a lot into a small genome.
Wow, overlapping instructions. That sounds like it needs very careful reading. And are these mitochondrial mRNAs different from the ones out in the main part of the cell?
Very different in some key ways. Most of them lack what's called a 5' leader sequence.
Okay.
And they don't have the Schindel-Garneau sequence you find in bacteria, which normally helps the ribosome find the starting point. So mitochondria needed to evolve totally different ways to initiate translation.
Ah, so no standard signpost saying start here. How do they manage?
We'll get into the initiation factors, but another feature is polyadenylation. Adding that poly-A tail at the end helps stabilize the mRNA, and sometimes it actually completes the stop codon, the signal to stop translating.
Clever. Okay, so unique mRNAs mean you need a unique ribosome, right? Tell us about the mitoribosome.
Yep, the mitoribosome. It's designated 55S, so it's a bit smaller than the 80S ribosomes in the cytoplasm. It's made of 2 subunits, the 39S large subunit, or MTLSU, and the 28S small subunit, MPSSU.
Still got the large and small subunit structure, though.
Oh, yes. The core catalytic part, the bit that actually links the amino acids together, is conserved from its bacterial ancestor. But layered on top of that are many unique adaptations and mitochondria-specific proteins.
Like what kind of adaptations? What makes it special?
Several things stand out. The channel the mRNA threads through is remodeled, likely to handle those leaderless mRNAs better.
Okay.
There are unique features on the large subunit, like a structure called the P-site finger, and changes to the L1 stalk region, which are involved in how tRNAs move through the ribosome during protein building.
And the exit tunnel, you mentioned the proteins are hydrophobic.
Exactly. The tunnel where the new protein chain emerges seems optimized for these hydrophobic polypeptides, helping them along. And crucially, the whole large subunit is actually anchored to the inner mitochondrial membrane.
Tethered. Why anchor the whole factory to the wall, so to speak?
That's absolutely key for something called cotranslational insertion. Because these proteins are so hydrophobic, they don't like being in the watery environment inside the mitochondria.
Right, they'd clump up or misfold.
Precisely. So as the protein chain is being made, it's simultaneously inserted directly into that lipid membrane. The tethering ensures the ribosome is right there, ready to feed the protein straight into the membrane where it belongs. It prevents aggregation and helps it integrate into the OXPHOS complexes.
Like a production line feeding directly into the assembly point, that's efficient.
Extremely. It highlights how specialized this whole system is.
And we mentioned earlier, problems here link to health issues. But thankfully, new tech is helping us figure this out.
Definitely. Things like cryo-electron microscopy, cryo-EM, let us see these ribosomes in incredible detail. And other techniques like mitoDMS-MaPseq and mitoribosome profiling are mapping out exactly what's happening on the mRNAs. We're learning a huge amount right now.
Okay, so we've got the unique landscape. Let's focus on initiation now, getting it all started. With those leaderless mRNAs, how does the mitoribosome actually find the right starting point? This seems like the first major hurdle.
It absolutely is. Initiation involves getting the ribosome assembled, bringing in the mRNA, finding the start codon, and recruiting that first special tRNA. It's complex.
And especially tricky without the usual signals.
Right. So mitochondria rely on specialized initiation factors. The main players are mtEF-Tu and mtEF-3. They're related to bacterial factors, but have evolved specific roles for these leaderless mitochondrial transcripts. They're like the specialist setup crew.
Okay, mtEF-Tu and mtEF-3, how do they manage the assembly?
Well, first you need to get the ribosome subunits together correctly. mtEF-3 plays a big part in preventing the small and large subunits from just sticking together prematurely.
A quality control step?
Sort of. It works with another factor, RBFA, ribosome binding factor A, forming these pre-initiation complexes. It keeps the small subunit ready, but waits for the right moment. There's another protein, MS37, involved, and some structural shifts happen that then allow mtEF-2 to come in. mtEF-3 physically blocks premature joining.
So it's a carefully ordered process, like building something step by step.
Exactly. You don't want the machine running before all the parts are in place and the instructions are loaded.
Okay, pre-initiation complex formed. How does it actually find the start signal on the mRNA and bring in that first amino acid?
There are a couple of models being discussed. One idea is that mtEF-2 brings in the initiator tRNA. That's the one carrying formamulocyanine, FMET.
The starting block amino acid.
Right. And mtEF-3 helps ensure that this tRNA correctly matches up with the start codon on the mRNA. mtEF-2 actually has a special domain to stop the tRNA binding too early before the mRNA is positioned. And interestingly, mtEF-2 seems better at grabbing the initiator tRNA than the regular methionine tRNA used later.
Why is that significant?
Well, mitochondria only have one type of methionine tRNA gene, but it gets modified differently for initiation versus elongation. So mtEF-2's preference might be an adaptation to make sure it specifically recruits the initiator form when needed.
Makes sense. What's the alternative model for initiation?
The other idea is a bit more like how bacteria handle leaderless mRNAs. In this model, the initiator tRNA, mtEF-2, and the mRNA all kind of assemble together onto a complete 55S ribosome that's already bound by mtEF-3.
So onto the whole ribosome at once?
Yeah. Then when the start codon is recognized by the tRNA, mtEF-2 burns some energy, hydrolyzes GTP, and this triggers mtEF-3 to leave. Once mtEF-3 is gone, the ribosome is unlocked and can start moving along the mRNA, making the protein.
Fascinating. Echoes of bacterial mechanisms still present. You also mentioned regulating the supply of the ribosome subunits themselves.
Yes, that's important too. The cell doesn't want a buildup of, say, small subunits if there aren't enough large subunits to partner with. So the levels of factors like MS37 and RBFA are controlled.
Balancing the inventory.
Exactly. And making the large subunit is also regulated. There's a complex involving proteins called male SU1, L0R8F8, and MTACP that acts like a chaperone during the final stages of MTL2 maturation. It helps fold the RNA correctly and form the active site, the PTC, while also preventing it from joining a small subunit too early. Only when this module leaves is the large subunit considered fully functional.
Checkpoints everywhere. Okay, subunits ready. How does the mRNA molecule physically get to the ribosome? Is there a delivery service?
It seems there is. The LRPPRC SLIRP complex is thought to be key here.
LRPPRC, SLRPS.
SLIRP seems to bind the mRNA directly, and LRPPRC interacts with proteins on the small ribosomal subunit, specifically MS39 and MS31. It essentially acts as a bridge, escorting the mRNA to the ribosome.
Delivering the instructions to the factory door?
Pretty much. There's even some evidence that a specific motif in the mRNA might have co-evolved with the MS39 protein, perhaps helping to stabilize that interaction. LRPPRC seem to guide the mRNA into the ribosome's channel.
And does LRPPRC affect the mRNA's shape or structure too?
That's been debated. The initial thought was maybe it just kept the mRNAs unfolded, but now it seems more likely that LRPPRC helps remodel the mRNA structure, maybe making it easier to process or load onto the ribosome, rather than just preventing folding altogether. It's probably involved in multiple post-transcriptional steps.
Okay, that covers initiation. Now, those bicistronic transcripts you mentioned, mtATP86, coding for two proteins, one after the other, with overlap. That sounds really complex to regulate. How does that work?
It's a fascinating piece of molecular engineering. mtATP8 and mtATP6 are two subunits of ATP synthase, the enzyme that makes ATP. They're needed in a 1-to-1 ratio in the final complex.
Okay, equal amounts needed.
But when researchers look, they find about twice as many ribosomes translating the first gene, mtATP8, compared to the second one, mtATP6. Yet the protein levels end up being roughly equal.
How? That sounds paradoxical.
It does. The key seems to be the overlapping region. It forms a stable stem loop, like a hairpin structure in the mRNA. And this hairpin sits right over the start codon for the second gene, mtATP6, effectively hiding it.
So the structure itself blocks the start.
In about 65% of the transcripts, yes. This structure is crucial for regulation. Upstream of the hairpin are these slippery sequences. The idea is that when the ribosome translating mtATP8 hits this hairpin, it pauses.
Okay, a roadblock.
And during that pause, there's a chance the ribosome slips backward by one nucleotide. This is called MADIS1 programmed ribosomal frameshifting, or PRF.
It changes its reading frame.
Exactly. And if it shifts into the MADIS1 frame, it very quickly hits a stop codon that wasn't there in the original frame. So translation stops, and you get a short, truncated ATP8 protein called ATP8-TR, which just gets degraded.
Ah. So many ribosomes start on ATP8, but a good chunk of them frameshift, make junk, and stop. That explains the higher ribosome numbers, but not higher protein output for ATP8.
Precisely. It's a way to make fewer functional ATP8 proteins than the number of ribosomes suggests, bringing the output closer to that needed 1.1 ratio with ATP6.
That is incredibly clever. What about translating the second gene, ATP6, then? How does that get started if its start codon is hidden or if the first round stopped prematurely?
This seems to involve mtEF-Ts again, that initiation factor. It's needed for translating the second ORF in both bicistronic transcripts, mtATP6 and mtND4. This points towards a mechanism called termination-dependent reinitiation, or TRA.
Termination-dependent reinitiation, so stopping one helps start the next.
Kind of. The idea is that after the ribosome finishes ATP8, either normally or after frame shifting and hitting that early stop codon, it terminates, but it doesn't completely fall off the mRNA.
It lingers.
Yes, the ribosome, or maybe just the small subunit, stays near the mRNA. Then it somehow gets repositioned or recycled to find the mtATP6 start codon and begin translating that gene.
How does it stay tethered? Does LRPPRC play a role here too?
That's the current thinking. The LRPPRC SLIRP complex might help hold the ribosome or the small subunit near the mtATP6 start site after termination of mtATP8 translation. This would make reinitiation much more efficient. Experiments where LRPPRC is knocked out support this idea.
So LRPPRC is like a master coordinator delivery and potential reinitiation assistance.
It certainly seems to wear multiple hats. And the fact that this whole mtATP6 start region is highly conserved across mammals suggests this PRF and TRA mechanism is an ancient and important regulatory strategy.
Really sophisticated control. Are there other places ribosomes might start translating on mitochondrial mRNAs besides the main known genes?
Surprisingly, yes. Using specific drugs that stall ribosomes right at the initiation step, researchers have mapped potential start sites across the entire mitochondrial transcriptome.
And what did they find?
They found evidence for potentially up to 31 cryptic initiation sites, places where translation might be starting at non-standard start codons, not the usual AUG.
31 extra start sites. Wow, what would that produce?
It would likely generate shorter versions of the known proteins, lacking the normal N-terminal end. For example, they found evidence for a shorter mtND5 protein starting from an internal site.
Do these shorter proteins do anything useful?
That's the big question. It's unclear if they have any biological function. It's possible they're mostly just aberrant products, mistakes. If they build up too much, they could potentially interfere with normal processes or burden the cell's protein quality control systems.
How might these cryptic starts happen?
One idea is related to that reinitiation concept. Perhaps when a ribosome finishes translating a main ORF, instead of reinitiating properly or dissociating, it scans a little and occasionally starts again at one of these nearby cryptic sites. mtEF-3 might even play a role in facilitating this by mistake sometimes.
Accidental reinitiation at the wrong spot. Okay, let's move on from starting translation to keeping it going elongation. What's the process there?
Elongation is the main cycle of protein building. It involves bringing in the correct amino acid carrying tRNA, dictated by the mRNA codon in the ribosome's A site. This is done by a factor called mtEF-Tu.
Okay.
Then the ribosome catalyzes the peptide bond, adding the new amino acid to the growing chain. And finally, the ribosome moves one codon down the mRNA that's translocation mediated by another factor, mtEFG1. Then the cycle repeats. There's also mtEF-Ts, which recycles mtEF-Tu for the next round.
So similar factors to bacteria and the cytoplasm, but mitochondrial versions.
Yes, the core factors are conserved. But again, the ribosome has specific proteins, ML40, ML48, ML64, whose movements seem to help shepherd the tRNAs through the ribosome during this cycle.
Are there any other mitochondrial-specific elongation factors?
There's mtEF4. It's related to a bacterial factor called EF4 or LEPA. Its exact role in mitochondria has been a bit debated, though. Some studies suggested reverse translocation, others ribosome rescue, maybe even roles in assembly or initiation. It's still somewhat enigmatic. Interesting. What about the protein exit tunnel during elongation? Right, as the protein chain grows, it passes through that tunnel in the large subunit. There's a specific mitochondrial protein, ML45, that sits near the exit. It actually has to retract or move out of the way to let the nascent polypeptide pass through.
A gatekeeper.
Kind of. It's proposed there's a gating mechanism involving movements of the large subunit, possibly linked to recruiting the OXA1L insertase complex, which helps insert proteins into the membrane. Remember that cotranslational insertion?
Ah, yes, linking elongation directly to membrane insertion via the tunnel dynamics. Very neat. Now, does elongation always happen at the same speed?
Definitely not. And this is crucial. The speed of translation isn't uniform, and pauses during elongation can be really important, especially for complex proteins like the hydrophobic ones made in mitochondria. Pauses can allow time for parts of the protein to fold correctly or to integrate into the membrane properly as they emerge.
So slowing down can actually be beneficial. What controls this speed?
In bacteria and the cytoplasm, things like codon usage bias, some codons are read faster than others, mRNA structure, tRNA availability and modifications, even specific amino acid sequences in the growing protein can affect speed. These are mostly unexplored territories in human mitochondria, but we're starting to learn about a couple of key mechanisms.
Okay, what have we learned recently?
Two main areas are emerging: control by the mRNA's own folding and control by the sequence of the protein being made.
Let's take mRNA folding first. How can the message itself control the speed?
Secondary structures in the mRNA, particularly stable stem loops or hairpins, can physically impede the ribosome's movement along the transcript. They act like speed bumps or temporary roadblocks, causing the ribosome to pause.
Like the one we saw in ATP86.
Exactly. But these can occur during elongation too, not just at initiation or overlap regions. And these pauses seem particularly important for proteins with multiple transmembrane domains, or TMDs, those stretches that span the membrane. Pausing might give each TMD time to insert correctly before the next one emerges.
Why would pausing be more likely in mitochondria?
Well, mitoribosomes lack some of the helicase proteins found in bacterial ribosomes that normally help unwind complex mRNA structures, so they might be more susceptible to getting slowed down by these RNA folds.
Is there evidence for this happening?
Yes, quite strongly. Researchers have found that hairpin structures in mitochondrial mRNAs often correlate with the sequence's coding for these transmembrane domains. MTCO1, cytochrome C oxidase subunit 1, is a key example.
What happens with MTCO1?
MTCO1 has 12 transmembrane domains. It's a very complex membrane protein. Analysis shows structured regions, potential stem loops, located around the mRNA sequences coding for specific TMDs like TM2, TM4, and TM6.
And do ribosomes actually pause there?
mitoribosome profiling data supports this. It shows ribosomes tend to accumulate, suggesting pauses, in regions corresponding to TM1, TM2, TM3, TM4, and just after TM6. There's even evidence of mitosome's 2 ribosomes potentially queued up behind a pause site accumulating after TM2 and before TM4.
Wow, actual traffic jams on the mRNA.
Pretty much. And other experiments using drugs have identified truncated MTCO1 proteins corresponding to lengths with 2, 4, or 6 TMDs having emerged from the ribosome, consistent with major pauses occurring after these points.
Why pause there specifically? Just for folding?
Folding and assembly. The pauses after TM4 and TM6 seem to coincide with the binding of specific chaperone proteins, COX14 and COA3. These chaperones grab onto the emerging MtCO1 protein, stabilize it, and help guide its assembly into the larger cytochrome 6 oxidase complex. Their binding might even help resolve the pause eventually.
So the pause allows time for helpers to arrive and guide the next steps. That links elongation speed to protein assembly.
Exactly. This idea of cotranslational and influencing elongation is a really exciting area. Since all these mitochondrial proteins are part of larger complexes, it makes sense that their synthesis might be coordinated with the arrival of their partner subunits or assembly factors.
How would assembly factors affect the ribosome speed?
It could be direct. Maybe the binding of an assembly factor stabilizes the ribosome in a pause state. Or it could be indirect. Perhaps the factor helps the nascent protein fold correctly, preventing it from getting stuck, which might otherwise cause the ribosome to stall permanently. It ensures smooth integration.
So the mtCO1 example with QX14 and COA3 fits this model.
Perfectly. The synthesis of mtCO1 seems tightly coupled not only to its own folding and membrane insertion, but also to the import of other cytochrome oxidase subunits made in the nucleus. And the timely arrival of these assembly chaperones like COX14 and COA3 during those programmed pauses. Some researchers even talk about a translational plasticity system allowing mtCO1 synthesis rate to adapt based on the availability of its partner proteins.
A responsive production line. Okay, the second control mechanism you mentioned was the nascent polypeptide sequence itself. How can the protein being made affect its own synthesis speed?
Certain amino acid sequences are just intrinsically difficult for the ribosome to handle. A classic example is stretches of proline amino acids, often appearing as polyproline PPX motifs.
Why is proline tricky?
Proline is unique. It has a rigid ring structure. Forming the peptide bond involving proline is chemically slower than for other amino acids. When you have several prolines in a row, this slowness can cause the ribosome to pause or even stall completely.
What happens during this stall?
The tRNAs in the ribosome can become unstable, making it even harder to continue. It can lead to ribosome collisions if others pile up behind, disrupting overall protein synthesis.
Do mitochondria have ways to deal with proline stalls? Bacteria and eukaryotes have special factors, right?
They do. Bacteria have EFP, and eukaryotes have EIF5A, which specifically help ribosomes get past these problematic PPX stretches.
Yeah.
But here's the thing. Mitochondria lack homologs of EFP and EIF5A.
Not necessarily. mtCO1, for example, is quite rich in PPX motifs, including a tricky triproline sequence. For a long time, it was a puzzle, but recently, a key mitochondrial protein called TaCO1 was identified.
TaCO1, what does it do? TaCO1 turns out to be crucial for relieving ribosome stalling, particularly at these challenging sequences like PPX motifs, but also others. Mutations in TaCO1 cause Lee syndrome, a severe mitochondrial disease, highlighting its importance.
So TaCO1 is the mitochondrial solution for ribosome stalling. Does it work during elongation?
Yes. Early ideas suggested it might be an initiation factor, but now the evidence strongly points to it acting during elongation. It seems to help ribosomes synthesize all mitochondrial proteins, but its effect is most pronounced for proteins like mtCO1, with those difficult sequences.
What happens if TaCO1 is missing or faulty?
You see significant ribosome stalling, specifically during mtCO1 synthesis. Protein levels drop, and you get an accumulation of shorter, prematurely terminated fragments. It shows TaCO1 is needed to get past those roadblocks. Interestingly, it also seems to help with pauses at some non-proline motifs too, suggesting a broader role in maintaining smooth elongation.
How does TaCO1 work? Is it like EFP or EIF5A?
The exact mechanism is still being worked out. It might function similarly by binding near the ribosome's E site, the exit site for tRNAs, and stabilizing the interaction between the tRNA and the catalytic center. However, some structural data suggests it might interact differently, perhaps near the A site and the L7L12 stalk region of the ribosome. There are bacterial homologs of TaCO1, and recent work suggests they might use distinct, though parallel mechanisms compared to EFP to resolve stalls. So mitochondria might have evolved their own unique solution with TaCO1.
Another layer of specialized machinery. And where do these proline motifs often occur?
Intriguingly, PPX motifs are often found near the boundaries of transmembrane domains.
So the difficult sequence itself might enhance those mRNA structure-induced pauses we talked about earlier?
Exactly. It could be a synergistic effect. The mRNA structure causes a pause, and the tricky PPX sequence makes the pause more profound or longer lasting, ensuring there's enough time for membrane insertion, chaperone binding, or assembly before translation races ahead. It synchronizes translation speed with these other crucial events.
Okay, synthesis is happening, pausing, assembling. Eventually it has to stop. Let's talk termination and recycling. How does the ribosome know when to finish?
Termination relies on stop codons in the mRNA sequence. When the ribosome reaches one of these, specialized proteins called release factors come in to recognize the stop signal and trigger the release of the newly made polypeptide chain.
Are stop codons the same in mitochondria?
Mostly, but with some key differences due to the slightly altered mitochondrial genetic code. UGA, which is usually a stop codon, codes for the amino acid tryptophan in mitochondria. And AGA and AGG, which usually code for arginine, act as non-canonical stop codons in vertebrate mitochondria.
So different stop signals require different reader.
Precisely. Mitochondria have release factors, MRFs, characterized by a specific GGQ amino acid motif needed for hydrolyzing the bond between the polypeptide and the final tRNA. But they seem to have distinct ways of recognizing codons.
Who are the players?
There's mtRF1A, which acts like a canonical release factor. It recognizes the standard UAA and UAG stop codons found at the end of 11 mitochondrial ORFs. It binds in the decoding center and interacts with the PTC to release the chain.
Okay, that covers the standard stops. What about those unusual AG code codons?
That's where mtRF1 comes in. It's responsible for recognizing these non-canonical AGA and AGG stop codons. The mechanism seems different, potentially involving a change in the mRNA's shape near the stop codon and recruitment of ribosomal RNA elements to help recognition, rather than direct codon reading by the factor itself.
An indirect recognition method. What about the mtND6 gene? Does mtRF1 handle its termination?
That's an intriguing case. mtND6 often ends with AGA or AGG, but getting rid of mtRF1 doesn't seem to drastically affect ND6 protein levels. This suggests there might be other ways to terminate ND6 translation, perhaps involving read-through or maybe other factors we haven't identified yet. It's a bit of a puzzle.
Always more questions. Now, termination isn't just about finishing, it's also linked to quality control, right? What if something goes wrong during translation?
Absolutely crucial. Stalled ribosomes or ribosomes translating damaged or incomplete mRNAs are big problems. Mitochondria lack some rescue systems found elsewhere, like translation.
So what do they have?
They have two release factors that seem specialized for rescue, related to a bacterial factor called ARF-B. These are C12 or F65, also called mtRFR, mitochondrial ribosome rescue factor, and ICT1, which is actually also a structural protein of the large subunit ML62.
mtRFR, what does it rescue from?
It seems to be involved in a pathway called mitoribosome quality control, mtRQC. It particularly comes into play when ribosomes stall because the mRNA is faulty, maybe lacking a proper stop codon, due perhaps to aberrant processing, like incorrect polyadenylation.
Why does it rescue?
While it has the catalytic GGQ motif, directly demonstrating its release activity in vitro has been tricky. But studies in cells lacking mtRFR suggest it releases the nascent polypeptide from ribosomes stalled on these nonstop mRNAs. It might do this by inserting part of its own structure into the empty mRNA channel.
So it detects the empty channel and triggers release.
That's one model. Stalling due to things like lack of available aminoacyl tRNAs can also lead to ribosome splitting, and mtRFR seems to interact with the stalled complex to release the polypeptide, while another factor helps remove the problematic tRNA. Cryo-EM structures show mtRFR associating with the large subunit when it's bound by that MLSU1 quality control module we mentioned earlier.
And the other factor, ICT1, ML62, it's part of the ribosome structure and a release factor.
Yes, it's weird. It has the catalytic motif and can act as a release factor for nonstop termination in vitro when it's isolated. But within the fully assembled ribosome, it's located quite far from the active site, PTC. It's unclear if it can actually perform release activity in vivo in its normal location, or if maybe a free form exists sometimes. Its precise role in rescue within the intact ribosome is still debated.
More mitochondrial mysteries. Okay, protein released. What happens to the ribosome itself? It needs to be ready for another round.
Right, that's recycling. After termination, the ribosome complex, subunits, mRNA, tRNA, needs to be disassembled. This is primarily handled by two factors, mtEF-G2, also called GFM2, and the mitochondrial recycling factor, mtRRF.
Not mtEF-G1 from elongation.
No, mtEF-G2 seems dedicated solely to recycling, unlike mtEF-G1, which does translocation during elongation. mtEF-G2 works together with mtRF to split the ribosome back into its large and small subunits, releasing the mRNA and tRNA. This step is essential to free up ribosomes for new rounds of translation.
Any other recycling helpers?
There's also GPPBP6, which is related to a bacterial factor called HFLX. It seems to function as an alternative ribosome recycling factor in human mitochondria, providing another route to disassemble the post-termination complex.
So multiple systems ensuring the machinery gets reset efficiently. We've covered a huge amount of ground here, from the unique setup to initiation, elongation control, termination, and recycling.
It really is a finely tuned process. Human mitochondrial translation depends on this intricate dance between the mitochondrially encoded RNAs and hundreds of proteins encoded by the nucleus, imported, and then assembled into this specialized machinery.
And the recent discoveries about mitoribosome adaptations, the roles of factors like LRPPRC and TACO1, the influence of mRNA folding, frameshifting, it paints a much more dynamic picture than we had before.
Absolutely. Seeing how translation is directly coupled to membrane insertion and protein assembly through mechanisms like programmed pausing is particularly fascinating. It shows incredible integration.
But there are still gaps, right? Things we don't understand.
Oh, definitely. How does this whole system respond to different cellular stresses or metabolic changes? How is it integrated with signaling pathways coming from the nucleus or the rest of the cell? And we've likely only uncovered some of the regulatory factors and mechanisms involved.
So future research using these advanced techniques will probably reveal even more layers of control.
I have no doubt. The deeper we look, the more complex and elegant it appears.
And understanding this better is key, given the links between mitochondrial dysfunction and so many diseases.
Precisely. Understanding how translation goes wrong is vital for developing potential therapies for mitochondrial diseases, and perhaps even for tackling aspects of aging or complex diseases like cancer, where mitochondrial function is altered.
It really brings us back to how fundamental these tiny powerhouses are. Thinking about all this complexity, the specialized factors, the RNA structures, the frame shifting. What's a final thought for our listeners to ponder?
Well, considering we've only recently uncovered things like programmed frame shifting and the role of TANCA1, it makes you wonder, what other completely unexpected regulatory strategies might mitochondria be using that we haven't even conceived of yet? And how could finding those change our approach to treating diseases linked to energy metabolism?
A tantalizing thought indeed. What hidden layers of control are still waiting to be discovered? That's definitely something to mull over. This has been incredibly insightful. Thank you.
My pleasure. It's a rapidly evolving and exciting field.
As always, don't forget to like the video and subscribe if you haven't joined us yet.